The brain is a fortress. It has to be. Guarding our thoughts, memories, and our very consciousness is a formidable wall called the blood-brain barrier. This highly selective membrane is brilliant at its job, keeping out toxins and pathogens. But this vigilance comes at a huge cost. For doctors and scientists, this barrier is a monumental challenge. A staggering 98% of potential drugs are blocked from entry, leaving devastating neurological disorders like Alzheimer's, Parkinson's, and brain cancers locked away, untouchable by our best medicines. But what if the key to unlocking this fortress wasn't a complex piece of human engineering, but a tiny protein found in the blood of a camel? Welcome to G Fun Facts Online. Today, we're diving into the world of nanobodies, a medical marvel poised to conquer brain disease. The story begins not in a gleaming, high-tech lab, but with a stroke of luck in the late 1980s. A group of university students in Brussels were working on a seemingly routine project, developing a test for a parasitic infection in camels. While analyzing camel blood, they noticed something strange. Alongside the normal, large antibodies we all have, they found a second, much smaller type of antibody. Decades of scientific belief held that antibodies needed 4 protein chains to work, too heavy and too light. But these camel antibodies were different. They consisted only of 2 heavy chains. This discovery, published in 1993, was a quiet revolution. Scientists isolated the smallest possible working piece of this unique antibody, and, because of its nanometer-scale size, they christened it the nanobody. To put its size into perspective, a nanobody is about 1/10 the size of a conventional human antibody. It's this tiny, compact structure that gives it a collection of biological superpowers. So what makes these nanobodies so special? It's their elegant simplicity. First, as we mentioned, is their size. Being so small allows them to penetrate deep into tissues and squeeze into nooks and crannies on cells that larger antibodies can't reach. Think of disease-causing proteins. They often have hidden weak spots or active sites tucked away in their complex structures. Bulky, traditional antibodies are often too large to get in there, but nanobodies can wriggle right into these cryptic spots and disable the protein. Their second superpower is robustness. Nanobodies are incredibly stable. They can withstand extreme temperatures, pressures, and pH levels that would destroy a normal antibody. This resilience makes them easier to produce, store, and potentially deliver in new ways, like oral pills or inhalers. Third, despite their small size, they don't compromise on performance. They are like heat-seeking missiles, binding to their targets with incredible specificity and strength. Their unique shape allows them to connect with targets in ways that other antibodies simply can't. And finally, because they are so similar to parts of our own antibodies, they are less likely to be seen as a foreign invader by our immune system. Plus, they're easy to engineer. They are encoded by a single gene, making them cheap to produce in large quantities using simple microbes, and easy to modify, like snapping together building blocks to create new tools. It's this unique combination of features that has taken nanobodies from a scientific curiosity to a potential game-changer for medicine. But the most exciting superpower, the one that could change everything for neurology, is their ability to cross the blood-brain barrier. How do they breach the fortress? One of the most promising strategies is a biological version of the Trojan horse. This is a process called receptor-mediated transcytosis. Here's how it works. The surface of the cells forming the blood-brain barrier is dotted with receptors, which act like delivery docks for specific molecules the brain needs, like iron. Scientists have cleverly designed nanobodies that can bind to one of these receptors, the transferrin receptor being a popular target. When the nanobody latches on, the cell says, "Ah, a delivery," and engulfs the receptor along with its nanobody cargo. It then ferries this package in a tiny bubble, called a vesicle, across the entire cell and releases it on the other side, directly into the brain. Researchers have proven this works. By fusing a nanobody that targets the brain barrier to a therapeutic molecule, they've successfully shuttled cargo into the brain. Another method, called adsorptive mediated transcytosis, is less like a Trojan horse and more like a magnetic key. The surface of the barrier is negatively charged. By engineering nanobodies to have a positive charge, they become naturally attracted to the barrier cells, which then pull them across. This allows some nanobodies to cross into the brain all on their own. These delivery strategies are the key that unlocks a whole new arsenal of weapons against brain diseases. Let's start with Alzheimer's, a devastating dementia that affects tens of millions. Imagine the brain of an Alzheimer's patient is getting clogged up with two types of toxic garbage: plaques of a protein called amyloid beta and tangles of another protein called tau. Nanobodies are being developed to launch a multi-pronged attack. Some nanobodies act as amyloid beta inhibitors. They are designed to find and neutralize the small, soluble, and most toxic forms of amyloid beta, preventing them from clumping together into larger, brain-damaging plaques. Other nanobodies are focused on tau. The spread of tangled tau protein through the brain is what closely tracks with cognitive decline. Scientists have developed nanobodies that can physically block tau from entering healthy brain cells, stopping the disease from spreading from one neuron to another. Beyond just stopping the problem, nanobodies can also be used as diagnostic tools. By attaching a radioactive tracer to a nanobody that targets amyloid or tau, doctors could use PET scans to see exactly where and how much of this toxic junk has built up in a living person's brain. This would allow for much earlier diagnosis and a way to track if treatments are actually working. Next up is Parkinson's disease, a condition characterized by the death of dopamine-producing neurons, which leads to tremors and movement difficulties. The main culprit here is the misfolding of a protein called alpha-synuclein. It forms toxic clumps inside neurons, eventually killing them. The challenge is getting a treatment inside the cell to deal with these clumps. Nanobodies can do this. A particularly promising nanobody, codenamed PFFNB2, has been shown to enter brain cells, bind directly to the alpha-synuclein clumps, and destabilize them. In animal models, this not only helped clear the existing aggregates, but also stopped the disease from spreading to other parts of the brain. It's a strategy that aims to halt the relentless progression of the disease at its root. Other nanobodies are being developed to target a different protein called LRRK2, whose overactivity is linked to Parkinson's, opening up yet another therapeutic front. Then there are brain tumors, with glioblastoma being the most aggressive and feared. Its prognosis is dismal, largely because it's so difficult to attack the cancer without harming surrounding healthy brain tissue. This is where the precision strike capability of nanobodies truly shines. Researchers have identified proteins, like one called ABCC3, that are found all over the surface of glioblastoma cells, but are mostly absent from healthy brain cells. They've developed nanobodies that can hunt down and bind exclusively to these tumor cells. Once they lock on, they can be used in several ways. They can carry imaging agents, allowing doctors to see the tumor's exact size and location. More importantly, they can be armed with a payload. Imagine a nanobody drug conjugate. A nanobody acts as the guidance system, carrying a highly potent chemotherapy drug directly to the cancer cell. It's like a smart bomb that detonates only on the target, maximizing its destructive power against the tumor, while minimizing collateral damage to the rest of the brain. An even more futuristic approach is photodynamic therapy. Here, a nanobody delivers a light-sensitive molecule to the tumor. When doctors shine a specific frequency of light on the area, it activates the molecule, triggering a chemical reaction that kills only the cancer cells. So, with all this incredible promise, are there any hurdles? Of course, this is cutting-edge science. One major challenge is that, on their own, nanobodies are cleared from the bloodstream very quickly, in just a couple of hours. That's not ideal for a therapy that needs to work over time. To solve this, scientists use clever tricks. They can attach the nanobody to a larger molecule that makes it too big for the kidneys to filter out quickly. Or they can fuse it to another nanobody that piggybacks on albumin, a long-lasting protein in our blood. This can extend its life from a couple of hours to several days. Another challenge is simply efficiency. While nanobodies can cross the blood-brain barrier, the amount that gets through is still relatively small. Researchers are working constantly to improve these Trojan horse and magnetic key systems to deliver a bigger therapeutic punch. And finally, like any new class of drug, nanobodies must go through rigorous testing to prove they are safe and effective before they can be approved for human use. The road ahead is challenging, but the future is incredibly bright. The discovery of Camelid nanobodies was a moment of pure scientific serendipity, but what's being built with them is a testament to human ingenuity. In 2018, the very first nanobody-based drug was approved for treating a rare blood clotting disorder, providing a critical proof of concept that this technology is not just science fiction, it is real medicine. Now, that same technology is being aimed at the brain. The tiny biological marvel born in the desert now stands at the gates of our most complex organ, promising to finally unlock a new era of diagnosis, treatment, and hope for millions suffering from the most devastating neurological disorders. Thanks for listening to G Fun Facts Online.